This experiment examines the theory that biodiversity provides critical ecosystem services that are important for sustaining agriculture in a changing climate.
The objectives are to measure the impact of biodiversity in time and space, as well as provide students with hands-on research experience.
The main plot examined winter cover and was divided into cover crop in blocks 1 through 3 and fallow bare soil in blocks 4 through 6.
The split plot examined crop diversity divided into 5 treatments: a corn, bean, and squash intercrop, corn, bean, and squash monocultures, and a fallow plot in each block.
Our group examined the crop environment in each treatment, collecting data on PAR, air temperature, relative humidity, soil temperature, and soil moisture.
PAR, or photosynthetically active radiation, designates the spectral range of solar radiation from 400 to 700 nanometers that photosynthetic organisms are able to use in the process of photosynthesis. This corresponds more or less with the range of light visible to the human eye.
Due to shading from taller plants, PAR will be greater at canopy level in monoculture plots for beans and squash compared to their canopy level in the Three Sisters treatment.
Air temperature influences rates of photosynthesis, respiration, and transpiration. Relative humidity affects the opening and closing of stomata, which regulates water loss through transpiration and photosynthesis.
Reduced air movement will result in higher air temperature and relative humidity within the canopy compared to above the canopy measurement.
Light transmission through the crop canopy affects soil temperature, which influences plant growth, and crop cover influences light transmission.
Soil temperature will be lowest in the Three Sister plots, slightly higher in monoculture plots with similar coverage, and highest in the fallow plots.
Organic matter influences soil properties, such as soil moisture, among others. Cover crops increase soil organic matter.
Due to higher organic matter in the cover crop plots, soil moisture will be higher compared to the non-cover crop plots.
Light intensity measurements are taken using a digital light meter. Place the light receptor in the spot to be measured, then wait for the meter to equalize, and press the hold button. Read the screen, first the large number, then the multiplier. Your partner can record this data. Release the hold button to prepare for the next measurement. Ensure the mode reads lux by pressing the mode button. When measuring in lux, you can convert to PAR by multiplying lux by 0.019. Light intensity measurements should be taken at the canopy of each crop and at the ground. In the intercrop, this means that measurements are taken at the corn canopy, then the bean canopy, the squash canopy, and the ground. Measurements should be taken on a line perpendicular to the ground so that all measurements are taken at the same point above the ground. In the monoculture treatments, only 2 measurements will be taken, one at the crop canopy and one at the ground. In the fallow treatment, only 1 measurement is taken at the ground. The spot where measurements are to be taken is determined by drawing a transect from one corner to the other within a plot. Light intensity is then measured at the 5 points indicated, which are spaced along the transect. Soil temperature measurements are taken using a soil temperature probe. The measurements will be taken along the same transect at the 2 points indicated. Make sure that the thermometer is in the ground when measurements are taken to be as accurate as possible. Air temperature and humidity are measured using a psychrometer. To begin, ensure that the cap is open by turning it counterclockwise. Then ensure that the temperature unit is set to Celsius. You can toggle the temperature unit by using the indicated switch. When the psychrometer is located at the site to be measured, close the cap by turning it clockwise. Press the hold button to pause the screen, then read the temperature and humidity for your partner to record. Press the hold button again to release the screen and prepare for the next reading. Air temperature and humidity are to be measured at the same 2 points as soil temperature were measured. These measurements are taken at both the ground level and within the canopy of the crop.
A loose measurement protocol likely resulted in variance between blocks.
Teams measuring different blocks on different weeks also contributed to this variance. A minimal amount of plant growth and a low population number in some plots made collecting environmental data near plants difficult and made any differences between plots low. A limited amount of time after the final data collection day reduced how much analysis we were able to complete before the movie date. When comparing soil moisture content between treatments, there was no statistical significance between the average soil moisture content of the cover crop plots versus the non-cover crop plots. There was also no statistical significance between the average soil moisture content of the intercrop and monocrop planting systems. We also examined the soil level relative humidity between cover crop versus non-cover crop plots and saw no statistical significance. There did appear to be a significant difference on day 3, although we feel this was probably due to a psychrometer error.
Some advice for next year. Figure out how you will collect your measurements prior to getting to the field. Break into 2-person groups for each block so that one person can measure while the other records. Have at least one of each tool per group. And keep your data sheets organized in the field so that data entry later is easier.